Yes, finally. So hello again. So just to remind you, where we stopped yesterday was thinking about, like now we've seen that Fisher's model might work like locally quite well, but now what about epistasis? So just to remind you, here's like Wright's fitness landscape with this topographic map with like fitness peaks and valleys. And so when talking about epistasis, I often ran into the problem that people actually understood something different than what I was saying. So I'm just going to tell you like the different definitions of epistasis and what I talk about when I talk about epistasis. So just looking at definitions, I found this one here from like the glossary of Nature Reviews Genetics, which says that an epistatic interaction is any non-additive interaction between two or more mutations at different loci, such that their combined effect on a phenotype deviates from the sum of their individual effects, which I think is a quite decent definition. And now let's look at an example just where you have like some specific phenotype and not fitness yet. So here is like Labradors, and so this is one of the, I think, a quite famous example of epistasis where you have a brown locus and which is recessive. And so if you have if you are homozygous for this brown locus, then you're gonna have a brown Labrador instead of a black one. And then there's a second one, and the second one is the so-called extension locus, and it's basically a locus that just takes away the dark pigment. And so what you can see is that if this one is also recessive, so if you're homozygous for the extension locus, then you're white. And so as you can see, so as soon as you have this extension locus, you're going to be white. And so there's an interaction between the extension and the brown locus. And actually there's something wrong about this picture here, because what mechanistically what's happening is that the pigment is missing. So in the extension locus, so in fact this dog here is actually, they put some like a wrong picture here, because it actually takes off the black pigment of everything, and so it should have a brown nose. But so you see you see kind of how it works, where you have like this interaction between the different genes. And on the other hand, there's also an epistasis definition from quantitative genetics, where in quantitative genetics you usually split up the variance in the population that for a trait that you observe into the genetic part and the environmental part and like the other fluctuations. But you can also put kind of split it up further and also put an epistatic part in. But most of the time this is actually ignored, because it's generally believed that on like long time scales epistasis doesn't matter, and so it's enough to actually only consider like the additive genetic variance in the population. And so when I talk about fitness to epistasis today, I'm going to use something that is more like the classical genetics definition of epistasis, but where the phenotype that we're looking at is actually fitness. So here, for example, we might have a reference type of like a reference strain of our yeast or so, and then there are two different mutations here, and so we measure their selection coefficient, and then we can get to an expectation what the combined selection coefficient would be if this purple mutation happened on top, or the green mutation happened on top of the purple one. And then if that is exactly what we observe, then there's no epistasis. If the combination of the two mutations is more beneficial than we would expect, then there's positive epistasis, and if it's more deleterious, then there's negative epistasis. So why should we care about epistasis? So one is that epistasis, due to this interaction, it actually can create non-random associations from a population genetic point of view. So it kind of distorts what I showed you at the very beginning yesterday, where you have, for example, just a clean like sweep trajectory, and it would also cause additional problems in the genetics of the population. And that is because it creates these associations that are described as linkage disequilibrium. But that's a relatively specific thing, but also this basically the epistasis describes how rugged a fitness landscape might be. So here's another picture of different fitness landscapes, And depending on the amount of epistasis, there is information on how predictable or repeatable evolution is. And so when I talk about predictable and repeatable in this respect here, that here's like just one type of definition, where, for example, if you have a very smooth fitness landscape, then it might be very predictable in the sense that you can always predict where it will end, the kind of your adaptive walk or your in this model. And then here in this case, it's also very repeatable because from those starting points, you also walk very similar ways to the optimum. Whereas if it could also be that you kind of wiggle around a lot, and so the path itself is not very predictable, like repeatable, but the endpoint is still predictable. And if you have a very rugged fitness landscape with a lot of epistasis, then it might be that it's neither predictable because you cannot just predict the endpoint, but also not repeatable. Although in general, what you might expect is that if you have a lot of fitness peaks, then it might locally be more repeatable because there's less different paths in the fitness landscape that you can take. And well, these kinds of things we would like to know, and they are very dependent on how much epistasis there is. And then finally, the accumulation of epistatic interactions is the basis of one of the maybe the most widely accepted model for speciation when populations are isolated. And I'm going to get back to that in a couple of slides to explain a little more. So, where do we actually expect to see epistasis if we look at genomes? And there are different possibilities. So for example, we could see epistasis within proteins just because in order to build this, build a protein structure, there are, just by definition, we even call them interactions. And so this should cause epistasis for fitness for the whole organism, because once you break the protein, I mean, that's the very simple example. Once you break the protein, your organism might be dead. But then if you add another mutation, you're already dead. So in a way, it's a very epistatic interaction. But those are maybe not the most interesting ones. But then there's also by definition, also epistasis between genomic regions that are involved in biological pathways, because if you just kind of for a similar reason, because if you disrupt the pathway somewhere, then you might have already kind of deactivated the whole pathway, and anything else might not add anything. And similarly, you could tune it in a non-additive way. And then, as I already said, you might also have negative interactions between species. And so, just to get an idea how you think about epistasis, here's my next question. So, where do you expect to see the most epistasis, and why? And so, we're going to have 2 minutes of discussions again between you guys, and I think this is everyone looks a little bit confused. Just let's see what happens. Yeah. Yes. So I was going to explain that later, but so you could imagine that. So if you have geographically isolated populations, they just accumulate genetic variants, right? So they will just over time neutral or non-neutral, or so they will diverge. And so just by chance, there can be negative interactions where something that evolved in one population is incompatible with the other, with something in the other. When they meet again, then you will see it, right? They will exist beforehand, and then once you bring them together, you might see those epistatic interactions. Okay, so let's discuss for 2 minutes. Okay, so for now we have to stop. We can still discuss this in the next few days, though, if you want to. So, does anyone want to share what they think, or shall we do a vote first? Okay, we can do a quick vote. So, who votes for within proteins? Okay, some people. And then the next one, the biological pathways. Also some between species. Okay. So can I get some opinions, please? Someone from the protein side.
Expression like color, and if that protein expression is dependent upon another protein expression which might inhibit the protein to express that particular green color. I mean, so in that sense, I would say epistatic. No.
Okay, but wouldn't that also wouldn't that also be a more like a between gene interaction?
Okay.
I mean, just I guess it could be both.
Probably. Yeah.
Someone else? Okay. Shall we do like, so who wants to vote for the, like who wants to go tell me an argument for the pathways?
I would expect that, say, if there's a signaling pathway where there are several components working, like depending on the activity of, say, the previous activator or whatever inhibitor, then if you disrupt at any one level, you would also disrupt, say for example, if you disrupt a transcription factor, you would affect the expression of the downstream locus as well. So that way.
And so that could actually also be quite prevalent, right? Finally, can I hear someone for the species argument?
DOBZHANSKY-MULLER incompatibilities, but I also don't want to explain that because I hope you have a slide for that.
Yes, it will come in a couple
But it's a theory that is used to describe why the biological species concept states that when things speciate, they won't, like when you try to bring them back together, they will not be able to produce offspring that are reproductively viable. And that's very confusing because if the whole process is just occurring neutrally, why is it that these species are not able to reproduce? So the DOBZHANSKY-MULLER incompatibilities uses epistasis between interacting, it's because of epistasis that you can still have neutral evolution that occurs differently between the two lineages, and this leads to incompatibility.
So I think that in a way this was a kind of bad trick question because firstly, I don't know the answer, but I would be very interested in hearing the answer and in thinking more about kind of quantitatively considering how many options we have to have epistasis, kind of to get a quick, like some kind of rough estimate of how much epistasis we would expect depending on those different levels. Oh yes, of course. I mean, and yeah, there are even more possibilities. And I think the other way how this was a trick question is that even like if you consider these incompatibilities that build up between species, this is like a non-mechanistic model, right? So you could get them and they fall also into those former categories, right? Which you will probably see in the following. But I thought it would be a nice kind of start of this discussion because I don't think that we know so much about how they are most prevalent, and now that people look more into where epistasis happens, it seems to be happening quite a lot, but still we don't really know how much it contributes to evolution. And I mean, for like a systems biologist, for example, it's absolutely clear that there's epistasis and that there's interactions also. Or like for Zorana, it's absolutely clear that there are interactions between things. but it's not, I mean, I don't think we have a clear picture yet how it on which time scale epistasis acts, and despite the fact that mechanistically we expect it to be quite frequent. So in the following, I'm going to guide you to some examples. And the first one I'm going to talk about is an actually within protein intragenic landscape that goes back to what I've already been talking about yesterday, this little region in the HSP90 protein. And so you remember what we did there, and that we had these 9 amino acids. And after studying like the single step mutations that I talked about yesterday, we wanted to look into whether we find epistasis if we make more than one mutational step. And so what we did was to take this wild type strain that we had before, and in this data set that I presented yesterday, we basically had 180 amino acid mutations. And so these are basically here like single step mutations, and all of them adds up to 180 and 3 replicates. And then we then built, we basically fixed 7 of these mutations separately. So for example, we picked this one here, And then on top of this first mutation, we made every other possible mutation again, such that we then had roughly 140 second step mutations for each of like 7 anchored mutations. So I'm calling this like the anchor. And then on top of the anchor mutation, we make the second step mutations. So that gives us roughly 1,000 or so double step mutations. And in that case, we did, of course, a biased choice, because if we picked a mutation that was highly deleterious, we would not get anything on top of it, right? Because it's very likely that nothing happens. So we chose these anchor fitnesses somewhere in the region of our wild type. And so then what we can look at are several questions. So here is just an example, like just to show you what kinds of things we might test. So if you now plot like the expected growth rate, if you just knew the single effect of the one mutation and the single effect of the other, how does this compare with what we observe when we make those double step mutations? And then we can go back to...
Can I interrupt? Do you know for a fact that the growth rate is directly proportional to the activity of the protein in question?
No. Yes. Yeah, we don't. I think we compared with what you can see, like at some point before I entered the project. I think they compared with some measures that you could look at, but then I don't think that they actually ever like measured this in our case.
Okay, it's an assumption though.
Yes. So then we could look at the distribution of fitness effects and how it changes when we do this anchoring. And so this is our old fitness distribution. So what we're basically doing is to take one step back, right? Like we're basically fixing this anchor, and then we can ask, okay, from this viewpoint, does it still look the same? Does it look different? Or like what is the shape of the distribution of fitness effects one step away? And then finally, we can also try to extrapolate and look at what is the shape of the local fitness landscape in two steps distance. And so firstly, now if we look at what happens epistatically, here is now our empirical measurements, where you have this expected growth rate on the observed growth rate, and it's basically colored. So these white areas are where we're really confident in our measurements. The crosses, they give us roughly like our posterior width, so our error expectation. And then this gray shade here corresponds to our measurement accuracy. And then these are the very strongly deleterious mutations, where we also don't have a lot of confidence in the measurements. But still, if you restrict yourself to these areas here, what you can see is that there is really a lot of negative epistasis, meaning that the double mutations are doing much worse in terms of growth rate than we would expect if there was no epistasis. And there are also a few that are positively epistatic, but in general, like there's almost 50% of the mutations that showed negative interactions. Oh, so we have the single effects, right? And so we basically add up the single effects, and that is our expect. Well, if they are growth rates, we multiply them, or we take we convert them into selection coefficients, and then we add them. So they are exponential growth rates, right? And so in that case, if you see them as an exponential growth rate, you would multiply in order to get your expectation. Okay, so you remember the slide here. We can now also, when we look back at the distribution of fitness effects, we again have a hypothesis that we can derive from Fisher's geometric model. And in that case, so yesterday, by having different environments, we basically moved the optimum, right? Because we introduced the population to a different environment. What we're now doing by fixing an anchor mutation is to actually change the location of the population that we are basing this distribution of fitness effects on. And so then the hypothesis arises that if we fix something that is deleterious, then in fact, we should get more beneficials to come up, right? Because we would expect that we move away from the optimum, and so we should get more potential for beneficial mutations. And we can test this by looking at the distributions of fitness effects. So here you see just a smoothened version of what I showed you a lot yesterday with this estimated growth rate now on the x-axis and the density of the smoothened DFE on the y-axis. And so the black line here corresponds to our original distribution of fitness effects. And then the colored lines correspond to the distribution of fitness effects of these different anchor mutations. And I think it's very, very easy to see that, in fact, we don't see a higher potential for beneficial mutations at all. So, in fact, what's happening is that we get a lot more deleterious mutations, and that's especially a lot of very deleterious, almost like lethal-like mutations, or stop codon-like ones. And suggests that maybe, and I mean, I don't want to go into this discussion so much, it could be that evolution took our reference strain to some kind of robust spot where single mutations might not matter so much, but then if you go 2 steps away, you might fall some kind of cliff in the fitness landscape. So this would result in something like a concave fitness landscape. And people have been studying different types of fitness landscapes that were like mostly biophysically motivated, for example, binding stability or like these kinds of things. And so we tested concave fitness landscape models, whether they could explain the pattern that we're seeing. And so in our case, of course, we have no clear phenotype that we can put into this model. So we just say, okay, so we have some phenotypic effect underlying the change that the mutation confers, and then this results in a fitness. And this relationship might be somewhat concave, such that you have like some optimum here, which is the light gray line, and probably our wild type or our reference strain is somewhere close to this optimum because it's well adapted, right? And because we're in the standard lab environment now. And so if you now make mutations randomly, then many of these mutations will be deleterious because you're already quite well adapted. And so imagine you have 2 mutations here, the green and the purple one, and then they are reflected in some kind of fitness on the y-axis. Now, if you do our experiment and you now combine these 2 mutations, so you put the green one on top of the purple one, then if you only look at the fitness dimension, then and you add them up here, you get to some kind of expected fitness of the 2 of them. However, if this is in fact happening on the phenotypic level, though, then this green one here will add up here with the purple one and might result in a quite different fitness that you observe in the experiment. And in fact, as you can see here, it results in an expectation where you have a lot of negative epistasis. And that seemed quite good. And so what we did was to then test like 2 different models that were like just kind of from what has been studied before in terms of concave fitness landscape versus our multiplicative model that we started out with which had no epistasis. And then here is an error measure, which is basically a Euclidean distance-based measure of how well we could fit the model. And as you can see, these 2 concave fitness landscape models, they did orders of magnitude better than our no epistasis model. Okay, so to summarize this intragenic epistasis part, we found that intragenic epistasis is frequent, and we're not the first people to discuss that. And so that what we saw was that changing this genetic background and dislocating the population from its current state did not result in a higher potential for beneficials. And so here we finally kind of cannot agree with Fisher's hypothesis, and we have to go to something else. And that's what we did. So we came up with this concave fitness landscape, which then could quite well explain the observed pattern of epistasis. Okay. So now let's get to a more global level of epistasis between species. And this is where we're kind of going back to your question. So this is the DOBZHANSKY-MULLER model. And so I did my PhD in speciation theory. So that's basically how I kind of started my my kind of dive into biology, and so it is widely assumed that this is a model that explains how you can get reproductive isolation in isolated populations. So imagine you split up like a population of some species and put them on different continents, and you just wait for a long time. So what's going to happen is that evolution just fixes mutations over time. And so you will get substitutions in each of these populations, and they might not be the same because it's driven, like it might be even just a completely neutral fixation of different mutations. And so over time you get these substitutions, and there's a probability that they are incompatible in the sense that they are negatively epistatic, because they have never been tested by natural selection before, since they evolved in different continents. And so what you might see is that eventually you get an accumulation of negatively epistatic alleles. And there's no expectation per se on what mechanism, like what underlying molecular reason might result in this. It's just a kind of a very intuitive thing that since natural selection has never been testing these combinations, they might be epistatic. And so that's the DOBZHANSKY-MULLER model. And in fact, many people have been looking for these DOBZHANSKY-MULLER incompatibilities. And there are already now many examples where they have been found. And so here's one of the examples, like a really classical one that I really like a lot. So this is a platy fish called Xiphophorus. And it lives in Mexico, and there are two different species here that have this specific DOBZHANSKY-MULLER incompatibility. So this guy here is Xiphophorus maculatus, and it carries a gene that is called TU here, and it's actually a tumor gene, but it also carries a repressor gene for that tumor. So what happens is that if you're homozygous for both, then or even if you're heterozygous, it's fine. So you will have black spots that are caused by this tumor gene, but you will be completely healthy. And in fact, what's going on is that females really like these black spots, so there seems to be some sexual selection for carrying the tumor gene. The other guy on that side here neither has the tumor gene nor the repressor gene. So when they meet and they actually do in nature, you see hybrids, and then in the first generation, hybrids are still fine because they are heterozygous for both of these loci. But if they now backcross to the guy that has neither the tumor nor the repressor gene, then you get some guys that have a tumor. In the second generation, you get guys that have the tumor gene but not the repressor gene. And in those cases, they still have black spots, but they will also develop fatal malignant melanoma. And so I think that's a really nice kind of example of how you really have this thing segregating in nature, where you have this very clear incompatibility between species. And I mean, it comes with all these also fun features, which are like the sexual selection that probably drove the tumor gene to high frequencies in the first time. And recently, they have done more studies on trying to find the interacting gene. And actually, it seems like that there is an actual like interaction with the promoter of the repressor gene, which is also a highly repetitive sequence. So it seems to be maybe also under selection. And what's also quite fun is that there is even ongoing hybridization in this species. So somehow, despite this strong negative effect, it still seems to stick around in those populations. And that was just a single DMI, and of course the first examples were usually finding like very strong ones because they were obvious in nature and much easier to test in the laboratory. So we have a lot of examples from Drosophila, for example, where they've mapped a lot of different incompatibilities. But nowadays you can also use other types of genomics approaches in order to identify interactions. And here's just one example where you see the mouse genome, and these are expression interactions, so-called EQTLs, and where they try to use quantitative trait loci mapping in order to identify interactions that are involved in reducing fertility in hybridizing mice. And all these blue lines correspond to potential interactions, and so you can see that the whole genome is somehow full with different types of interactions. And so, so just to highlight this again, I mean, both of these examples that I told you, they are in fact also biological pathway stories, right? So, and that kind of leads back to what I was saying in terms of my trick question that I asked you earlier, that of course, like that, what this kind of more phenomenological model of the DMIs could actually be quite, is almost, I mean, how to say this? So it basically proposes just how this intuitively happens, but then if you look into the existence of pathways, and so it's also a very intuitive way to just mechanistically justify why this should be happening. Okay, so now in the final part of my lecture, I'm going to take a look at like whole fitness landscapes. So here's one of the first whole fitness landscapes that people have mapped, and I think yes, we will. Yeah. So we have already heard from I don't remember. Somebody mentioned the Lansky long-term experiments, right? Where Rich Lansky, he took those 12 E. coli lines and he put them into culture 30 years ago, and then has been propagating them for now like 30 years and maybe 70,000 generations, and just in a constant environment looked how they adapt to this lab environment. And so in 2011, they took one of these lines and they actually identified the 5 first mutations that fixed in those lines, and they wanted to know, okay, so how did these mutations interact? Was there a lot of epistasis? And so what they did was to artificially create the fitness landscape of all combinations of these 5 mutations. So here on the top is the ancestor, and on the bottom is the mutant that has like the genotype that has all 5 mutations that fixed. And so the true actual path that it took was, I think, the one. So I think this is the order in which they appeared. And now you can see how the relative fitness, which started, of course, at 1, right? So that's our reference point. And then this is the fitness of the fittest type. And what you can see is that so the solid lines represent paths that actually lead to increasing fitness. And then the dotted lines, they represent paths that decrease the fitness of this different type. And so as you can see, then in this fitness landscape here, it actually looks quite smooth. So there's a lot of solid lines, meaning that many of the paths in this fitness landscape were accessible. And there's in fact only one, like a single fitness optimum, which is actually the one that was reached in the actual experiments. And so when you look into the selection coefficients, and that's of course, so that's not visible in this picture here, which is just shaded, was that there was a lot of diminishing returns epistasis, which means that the first mutation still had kind of large effects, but then if you add another mutation, then the effect of the next mutation was always lower than expected by just adding it to the one that came before. And that is what is called diminishing returns. And so that's quite interesting because it was both accessible in the sense that you could reach this single global optimum quite easily, but it was still epistatic. And so this is just one example of a fitness landscape. So now just a quick kind of way back to like why are these fitness landscapes so appealing? And so far, I mean, we've been going like stepwise basically to this big fitness landscape and discussed like models, that different types of fitness landscape models. But if we now have these big genotypic fitness landscapes, then actually we could test all of this at the same time, right? Because, so I've already argued that you could test the predictability and repeatability of evolution. And also, in a way, it becomes increasingly simple to measure these experimental fitness landscapes. And we should also get a lot of expectations on epistasis just by the accumulating data that we have on gene networks and pathways. And so basically then like this whole combination has led to almost like an explosion of fitness landscape studies in recent times, where it's like almost like it's kind of this yeah, they are pretty much everywhere, and I sometimes, I get very depressed when I look at the new papers that come out and I see what I should all read about fitness landscapes every day. And so, however, we still have a lot of problems also with these fitness landscapes. Because the problem is, firstly, they are enormously complex, right? So when you look at Wright's fitness landscape, it's just this topographic map. So you think you can just, fold it up and then you take your path and so, but that's not really true for a genotype fitness landscape that has actual genotypes on it, right? It's a multidimensional hypercube. And so even with the best experimental methods, it will not be possible to ever map the whole fitness landscape of like even a whole gene, right? And then also the question is, do we really need all this information? Like if we had this big fitness landscape, what would it tell us really about evolution? And can we then predict anything if we have this type of fitness landscape? But still, I mean, I'm still relatively optimistic because I think that if we find the right ways and the right models, then maybe it can help us to predict things like, for example, the cause of antimicrobial resistance if you want to apply it to something. Anyway, so now we are going to go back to the experiment for the last time, and you already remember this. And just to summarize how we went through this, so we started with these single mutational steps, where we then looked into how this distribution of fitness effects changes across environments, and then we also looked at what this tail might tell us about adaptation. And then we also took the second step and looked at how epistasis works out if we add a second step to our experiment. And so now in the very last part, I'm going to look at like a full or as full as possible fitness landscape of many combinations of mutations still in that same area, and look at what it might tell us about evolution. And so just to tell you what this data set is, so we're back in the high salinity environment, and that is because we also wanted to include some mutations that might be beneficial. As you remember, in the standard environment, we found almost no beneficials, right? So it made more sense to actually look into an environment where we can find beneficial mutations. And then we picked 13 of the mutants that we had in our original data set, and we built every possible combination of each of these mutations. And that would already be a lot, but it's slightly reduced because we actually picked mutations at the same amino acid position. So the maximum number of mutations is 6 at the same time, right? So these are always up to 6-step mutations. And what was kind of nice about the data set was that we then had these multi-allelic fitness landscapes where we could look at what different alleles at the same locus could change in the fitness landscape. And so that results in roughly 1,600 nucleotide mutations, but we reduced our analysis to the amino acid level, so we ended up with 640 different genotypes. And so, in a way, this is what we did was a relatively unbiased selection of mutations. So one of the problems with fitness landscape studies is that you can't do as I already said, it's really hard, like you can't literally do the whole, not even the whole region of 9 amino acids. So we had to choose some, and we tried to choose some kind of selection. And of course, we didn't select anything that was very deleterious, but we had some mutations, including one that was a little bit deleterious and some that were not distinguishable from wild type. So yeah. And then, yes. So then we asked 2 questions in this study. So the first one was, if we only knew the single effect mutations, could we predict how we can go to the optimum in the fitness landscape? So kind of going back to the earlier question, if we had only this distribution of fitness effects from the beginning, would it be enough to tell us where we can get if we combine several of them? Then the second one is, if we now have our fitness landscape and we simulate evolution on this, will it actually take us to the optimum? So how accessible is this fitness landscape? And then finally, we asked whether it's possible to infer an unknown part of the fitness landscape and whether we can extrapolate from a smaller part to the bigger landscape. So just a quick look at the whole fitness landscape. So here, now on the x-axis is the mutational distance from this parental sequence that we have always used as a reference type. Here, back here is, so on the y-axis is the growth rate, and so we start at our reference type, and then we have up to 6 mutational steps. And so, yeah, this is basically what you see. And so what you can see already is that there was a lot of beneficial mutations, but also a real lot of deleterious mutations. And so you see, for example, also vertical lines, and these are mutations that happen at the same locus. So these are the multi-allelic ones, right? Because they don't change the distance to the wild type, but they still are a mutation in the fitness landscape. And then this guy here was just a control where we put in a stop codon to see what roughly is something very deleterious. And so you can see that there, because this goes like this, there's obviously already a lot of negative epistasis in the landscape. But getting to the first question, now if we think of the single-step mutations, if we only knew the single-step mutations, which were these guys here, do they predict the way to the global optimum? And so what we focused on were 3 specific landscapes. So here is the whole landscape again, and here's just the part that was beneficial. And the yellow one would be that fitness landscape, so the global optimum that we found, so the highest fitness genotype in the whole landscape was 4 steps away from the reference type. And so what we picked first was one fitness landscape in which we just picked the 4 mutations that actually lead to the global optimum. And so we looked at whether the 4 single-step mutations that are involved in this sub-fitness landscape actually predict where you can get with the 4th step. Then the second one we picked was the one that had the best 4 mutations. And this means that we picked the ones that had the highest selection coefficients among these single-step mutations. And then finally, we picked the ones that had the worst 4 mutations. And so, of course, we have only 1 true deleterious mutations in there. So I put this in, it's not really deleterious mutations. It's just the ones that had the lowest fitness effect in our data set. So what we can now look at is the actual realized fitness effect of those combined mutations versus the expectation that we had. And so here is the yellow one is the one that actually leads to the optimum. And this is what we expected from just adding up the selection coefficients of the single-step mutations. And what you can see is that we clearly expected that it would be beneficial, but in fact, the optimum was much higher than we were actually thinking. So there was a lot of positive epistasis between those mutations that lead to the global optimum. Now, the picture was very, almost opposite when we picked those other two fitness landscapes where we combined all the best mutations and all the worst mutations. Because what happened there is that these two were the expectations when adding them all up, but what we actually saw when we compared them was that they were much, much more deleterious. And so, in fact, by just combining the most beneficial ones, we would drive the fitness over some kind of cliff and end up in a very deleterious part of the fitness landscape. So to sum this up, we saw both strong positive epistasis on the way to the peak, but also very strong negative epistasis everywhere else. And in fact, so this positive epistasis on the way to the optimum, we didn't only see for the global optimum, but we also saw this for any local optimum in the landscape that there was positive epistasis. Okay, so now here's just a different picture of all the beneficial mutations in the landscape, which are now colored by their fitness. So this is the reference type, and this is the highest fitness type. And here's our parental genotype, and here's the global optimum, which has this red square. And then all the local optima, they have orange squares. So just to make it more clear, we had in this beneficial part, we had 5 local optima, and then the gray arrows, they always point towards increasing fitness. And we can now ask how accessible the landscape is and whether we expect evolution to actually lead us from the parental type to the global optimum. So what we can do for this is to basically simulate evolution, or even kind of write it down analytically, the probability from any genotype in the full landscape to actually go to the optimum. And that's the way this works is to basically assume that we have weak mutation strong selection limits, such that only one mutation happens at a time. And then this mutation is fixed with a probability that's relative to its selection coefficient as compared with the other beneficial mutations that you can reach in a one-step distance. And so you basically can start somewhere, and then you look how many more beneficial mutants there are in one-step distance, and so you can then calculate the probability to go to that point, and then you will see which ones are the absorbing states and how what the absorbing probability is from any point in the fitness landscape. And what you end up with then is this type of plot here, where this is the absorbing probability. So this is the probability of ending up in this global optimum from any point in the landscape, and how often this probability is reached. So what you can see here, for example, is that on average, there's a lot of this optimum here is very accessible. So there are many places in the landscape where you're very likely to actually go to the global optimum. On the other hand, what's quite interesting is that...
Sorry, it's me. Can you explain what an absorbing probability is again?
So it's the probability that if you start somewhere in the landscape, that you actually go to the global optimum. So basically, each data point here is a starting point in the landscape. So there are basically 640 data points summarized in this histogram. And for each of them, we write down what the probability is that it goes to this point here, and then we count them all up. And that's kind of, so for example, this, like the 1 here, means that if you start in that point, in one point in the fitness landscape, you will always go to global optimum. And that is, for example, always true if you're very close, right? So if there's nothing else around and you're like just on the way to an optimum, then you will, with a probability of 100%, go there. And that happened actually quite often in 2% of the cases. So in 2% of all initial starting points, you would have a 100% probability of going to the global optimum.
So we have obligatory paths. In some cases, there are obligatory paths.
Yes. Mm-hmm. And I mean, a very intuitive one is that if you're already there, you can't go anywhere else, right? Because you're in the global optimum, so you have 100% probability that you will evolve. So that's a trivial one. And also another trivial one is if you're in any of the local optima, you will also stick there because there's no other way out. But then there also seem to be others where you get on a path that only can lead you to the global optimum. And now just to compare, if we started in the reference type, we have on average a lower probability to end up in the global optimum, which is only around 25%. So you can also go kind of around because they are multi-allelic landscapes, so you can kind of mutate several times at the same locus, and so it's just that once you're in an optimum, you're going to stop. So it's a probability that's relative to your selection coefficient. So you assume that every mutation is equally likely, but then it's weighted by the relative fitness gain that you can have at this position. And so what this shows us here is that even though on average the fitness landscape is quite accessible from this reference type, which is where we would start an evolution experiment, for example, it's relatively unlikely to actually get to that high fitness point. And to compare this with another local optimum for this one here, for example, which is only 2 steps away from the reference type, we see that on average, the probabilities of reaching this optimum are much lower, and that's also because it's less fit, right? And so the ending fitness in this local optimum is lower, but then the probability of going there from the parental type is much higher, and that is partly due to the fact that it's only 2 mutational steps away, right? Because if you're already quite close, then and there are some accessible paths, then it's also more likely to reach this. So you would reach this one here with almost a 50% probability, and then you will be, in the case that you have no drift or so or anything happening, you will be stuck in that local optimum. And so what this told us about this particular fitness landscape is that on average this guy here was very accessible, but it would not necessarily be what's actually reached in experimental evolution approach, where you would just put your reference type in a new, in high salinity and, you know, let it evolve to get to somewhere. And I mean, just to be honest here, this, of course, this is I think it's a very nice exercise to understand how fitness landscapes work. The problem is that none of these mutations are really present in nature, right? So this is a very, very conserved region. And I think one of the difficulties is now to understand whether we can actually extrapolate to like nature from this, right? Because even if we put this reference type into high salinity, it might be that anything can happen somewhere in the genome, but we have only looked at 9 amino acids, right? And so I think one of the difficulties I personally have when trying to interpret these things is that it's a really very nice nice framework, and you get a lot of information out of it, but it's not absolutely clear what this teaches us about evolution in nature. Well, I mean, here we just didn't consider any population size or so, right? So this is more like a Gillespie type model, which is just a strong selection, weak mutation limit, where we, for example, where no slightly deleterious or neutral mutation can ever fix, right? So you only will go upwards in the fitness landscape. So in that sense, in spirit, it's more of a Fisher type model, because it's very strongly based on the assumption that you can only increase your fitness, rather than, you know, being able to traverse between fitness valleys. Okay, so that tells us if we believe that we can extrapolate to natural populations, then this would mean that it might be quite likely that in such a case, evolution from this reference type may stall at this local optimum. And so finally, we looked into different types of describing the landscape and whether they would be helpful to extrapolate beyond like a known part of the fitness landscape. And so far, when I talked about epistasis, this was always measured based on the reference type, right? But that is a very biased measurement, because depending on which reference type you choose, you have very different values for your epistasis. And so we looked into this new measure of epistasis that was proposed in 2016, which is not based on any reference type, but it's based on the correlation of fitness effects across different genetic backgrounds. So just to explain you this here, this is the fitness landscape again, and now we can just pick two mutations, and basically starting here and making a tiny fitness landscape in this. And so the blue mutation has a beneficial effect on its own, and the purple one has a deleterious effect. And now if we have the blue one first and then the purple one, it still has a deleterious effect on top of the blue one, so there's no epistasis. And the same is true for the blue one on top of the purple one. So this is a perfect case of no epistasis. And it also means that the fitness effects are perfectly correlated across the landscape. And that is basically this measure that Guillaume Achard and his people proposed, which just looks into the correlation of fitness effects across the whole landscape. And then you can do this based on every mutation, every genotype in the landscape, average over all of them, and that will give you some information about how much epistasis you have in the landscape. And just to give you a different example here, for example, the blue one is by itself deleterious, the purple one is also by itself deleterious, but the purple one on top of the blue one is beneficial, as is the blue one on top of the purple one, and that would be a case of very strong epistasis. And in fact, now these two mutational effects are anti-correlated. And that way you can already see that if you have like a perfect correlation between fitness effects, you will have no epistasis. If you have anti-correlation of fitness effects across different backgrounds, you have a lot of epistasis. So what you can then do is to build something like a fingerprint of your fitness landscape. So now here you see any possible combination of substitutions in the landscape. And then in color is basically this fitness landscape, this correlation measure. And if it's dark blue, then there's no epistasis. If it was red, which it never is, then there would be very strong epistasis. And so you have to keep in mind that this is averaged over the whole landscape. So this means that now if we have like this mutation and this mutation here, then this means that this combination of those two mutations on average over the whole landscape has this type of epistasis. And that also results in the fact that we don't really see a lot like strong epistasis all the time because it really has to happen across the whole landscape. And what you can also see is that it's asymmetric because whether one mutation happens on the background of the other or the other one on the background of the first is, or of the second, actually can give you different types of different correlations. And so that means that you can measure it both ways. And now if you stare at it long enough, then you will see that there is actually some kind of pattern that you can see in the landscape. Even though on average there's not too much epistasis, there's clearly some mutations that stick out here. And these are all the ones where something mutates to a proline at position 588. And so we looked into that mutation, and what it actually is that this proline mutation, it somehow locks the conformation of the protein such that any mutation that follows is much more likely to have a deleterious effect. So that was really nice because it could basically, it showed us that this measure of this epistasis measure can give us actually some information about what is going on in this landscape, and it agrees with something that we also know has like a biochemical or biophysical basis. Okay, and so finally we now looked into this epistasis measure to see how much information it gives us to extrapolate beyond the known part of the fitness landscape. And so I mean, so far I've shown you this big fingerprint, but there's a way to actually summarize this gamma statistics and then to make it depending like on the average gamma across mutational steps in the landscape. So it's almost like a measure of like first order epistasis up to distance, or like one-step distance epistasis up to five-step distance epistasis. And in that paper, they looked into basically the signature of different theoretical fitness landscapes that they would leave when you look into the statistic. And I'm not going to go into detail explaining what all these models are, just a little bit here. Like this red line is an additive model where you have no epistasis, and clearly there's like this measure is 1 for all these, for any distance or in the landscape. And then you have one that's very interesting, like that's the house of cards model, where you have kind of like an absolutely random landscape. So in theoretically, epistasis is maximal. And then one that's also very, very famous is the NK model. And so now you can basically calculate this for your empirical landscape, and you can look into what it looks like and which type of landscape it resembles most. And so this is for our landscape, where we have this blue line that corresponds to the global landscape. And so on average, it looks like something that could resemble this what's called RMF here, which is a rough Mount Fuji model, and that's just a tunable type of fitness landscape somewhere between House of Cards and additive. And so it looks like, okay, that could be something. And what we now asked was, if we now exclude mutations from the landscape, so we look at a smaller part of our fitness landscape, does it still look the same way with respect to this signature? And so in this plot here, what we did was to just drop a single mutation from the fitness landscape. So the gray lines correspond to dropping any other mutation, and then the purple one corresponds to dropping just this one mutation, which I told you in the slide before, it was this like epistatic hotspot mutation. And so you can already see that on average, if you drop one mutation, you still recover this signature of the fitness landscape. But if you drop this one, which we have shown to be quite epistatic, then there's already a little bit of a different signature in the landscape. And that gets even more exaggerated if we now don't look at this multi-allelic landscape anymore, but if we look at every sub-landscape that has 6 different mutations. So we have, like, I told you that we have up to 6 mutations, but so far at each locus we could have several, like at some loci we could have several mutations. So now we restricted to only 2 mutations, or like one mutation at each locus. And even already then you get a really large spread of different signatures with respect to this the statistics, where this is our whole landscape. And then now you can just pick a couple of different ones. And so the green ones are all those that I think in that case have the 588P mutation. So already here, a sub-landscape might not tell you much about how the big landscape looks like, which indicates that there's like local heterogeneity in the landscape that's not really covered if you just describe the whole landscape. And yeah, it becomes even crazier if you now restrict it to 4-step sub-landscapes where you get any kind of pattern. I mean, it's not visible very well, but you get anything kind of in between, and so every possible type of fitness landscape that you might imagine. And so to summarize this part about the big fitness landscape, on average, we saw that our intragenic fitness landscape is quite rugged and that negative epistasis is common if we use this other epistasis measure that I introduced at the beginning. And the global peak is accessible, and it is always reached through this synergistic combination of 4 mutations. But we're really far away from, oh yes, but when we assume that there's evolution from the parental type, then we might think that adaptation could get stalled at the local peak. And what I wanted to say is that we're still very far from figuring out how to use these fitness landscapes, like pieces of the fitness landscape, to predict anything beyond what we actually observed. And so just to kind of finish up here, so I think there are still many open questions, right? And now if we go back to our question, like, okay, Fisher or Wright or something, I think there's still a lot of potential to study different ways of describing like adaptation and quantifying epistasis and so, and also figuring out what actually affects evolution, I mean, even if we had, like, if we knew everything about molecular interactions and so on. And so one thing that I think is important is that so far, I mean, I've always talked about stable environments and like very, very easy kind of, or like maybe for me as the theoretician, easy experimental, kind of very clean experimental setups. But then there's also like the ecology and demography playing a huge role. And in fact, I think from like, to go back to like our Wright question, I think that also like bottlenecks might be happening very often. And this picture of just going upwards all the time might not be so true after all. And then even if we ignore all of this, still it's a super exciting, I mean, if we look at adaptation, for example, in the long-term experiments, it's still very miraculous how it actually happens, and we know still like there's so many open questions. And so one thing here, here's just one plot where this is again this long-term experimental evolution experiment over 50,000 generations, where they measured, found some kind of measure of relative fitness, which would tell you how the fitness increased in those Lansky lines. And what they saw is that, I mean, any kind of model that we talked about had some kind of optimum, right? Like be it the Wright model, where you just have some kind of global optimum once you know the whole landscape, or like Fisher even more, where you might even have a single peak, or like our biophysical model also had like a converged to some kind of optimum. But what they saw is that even after now even like 70,000 generations, it doesn't seem like adaptation is actually stalling, so you're still increasing fitness. And one of the questions is really why this is happening, and I think two of the hypotheses that have been invoked to explain this is firstly that there could be so much epistasis that you really have to find your way, even in this population, and there are still some rare occasions where you can optimize. But then one thing is also, again, like the ecology, where it could be that the organism feeds back onto its environment. And so what we always assume to be a stable environment, because it's a very defined, well-defined medium and whatsoever, is actually not so stable after all, because just evolution itself changes the environment due to the organisms inside the experiment. And so this is maybe just the final kind of contrast with what I've shown you, where we tried to really make it very simple and very stable and very kind of based on like selection and kind of more or less predictable or so. And I think I mean, I'm very excited to think about these things, and I think it's important to take all kinds of different approaches, but it's important to keep in mind how complex and how exciting nature actually is when you look into these things, even in the lab. Okay, so my last question for you today is, so what would you like to know about fitness landscapes after those 2 days? And I'm going to give you 2 minutes again, and then we can more or less finish up after a little bit of discussion. Please discuss between like 2 people for 2 minutes, and then we can later have a group discussion about it. So no one has questions anymore? You must be discussing something. Okay. So before I finish up, I would just like to hear maybe like 3 people kind of saying what they would like to know.
I'd like to know what the appropriate axes are. Frequency, genotype, or phenotype.
That's a wonderful question. Somebody else? How to measure fitness? Also a very good question. And I've completely ignored that throughout the last 2 days, pretty much. Yes, very good. And one more. Oh, what's the fitness of a heterogeneous population? Mm-hmm. Yeah, that kind of adds up to this one and makes it even more complex, yes. And I think, yeah, these are great questions. So maybe just to wrap up here, firstly, if you want to learn more about evolutionary dynamics and see some cool speakers, we're going to have a conference in October, and now registrations just opened. We're going to have a great lineup of speakers, and it's partly organized, including by lab members of mine, including Hermina. And so you can ask her everything about the conference. And with that, I also want to thank everyone, like my collaborators in particular. Without them, like all my work would be impossible, and my lab, which is really great to have around, and like some funding people too. So thanks, and I'm happy to take more questions.
So again, I'll give a minute or 2 to the audience to formulate their questions. There are many questions I'm sure they have, and I'll start with my question, which is back to obligatory paths or near obligatory paths. Do you think that's a good argument for the crucial importance of big genome rearrangements, in the sense that they just reset the initial condition, which would suggest that looking at SNPs just in the context of SNPs might be of little relevance, and that would also suggest that evolution, when having only the tool of S&P is just extremely constrained.
So I think that this, like our idea of having just really like single mutations that have like a single, maybe even non-epistatic effect is a very simplified way of thinking about things. And I totally agree with you that we're very far from understanding and also modeling these things. I mean, there are a lot of arrangement models already around, but I think that the future will be to actually also kind of include this. I mean, I personally am very excited about including this type of thing into fitness landscape models where you can somehow figure, like you can think of ways where like a single mutation might completely change the local fitness landscape. And I agree that this is something that could change like also explain a lot of things and also change the way we think about these accessibility questions.
I think that one assumption of fitness landscape is that like all combinations of traits are possible. And so I would like to know if there are some works when you show the landscape, but showing for example, genetic constraints. Like for example, we can have this theoretical model where 1 or 2 or 3 mutations are possible, but maybe some specific combination are not just possible. Like for example, one gene that inhibits the other, you will never find both genes expressed, say something. So I don't know if there are some landscape with those, maybe also physical constraints and stuff.
So I mean, in a way, the concept of fitness landscape would include this, right? Because those combinations that you're talking about, they would just end up being very deleterious. But I think one of the steps to do is to extract information from biological pathways, for example, and figure out ways of mechanistically bringing them to some kind of fitness landscape and figuring out like what they like, which type of epistatic pattern arises just from knowing these constraints that you were talking about. You look like I didn't answer your question.
No, yeah, maybe with, yeah, I don't know. I have to think it like,
okay, well,
strange, I don't know why, maybe they have great fitness, but they're just not possible.
So you mean mutationally, like that this mutation just won't happen?
For example, or no, maybe like physical constraints, like on form and like some possible theoretical forms that are just not possible in nature due to physical constraints, and so
but then in a way it would still be accounted for by just giving them zero fitness, right? Because whether they occur but never make a difference or whether they are not possible is like from this theoretical point of view, I think indistinguishable.
Okay.
So, yeah.
Okay, thanks.
Okay, I think we can probably continue discussing also over the week, right? So, okay, thank you.